After studying this module, you shall be able to know about the significance of drug extraction, the basics of drug extraction, and the different procedures and techniques used for drug extraction. The process of drug extraction is the principal step in the qualitative and quantitative analysis of drugs from complex biological specimens. The large groups of substances that are relevant vary greatly in their physiochemical properties. They are time and again potent agents that are present only in very low concentrations. Therefore, a chosen extraction procedure must not only be capable to separate the target substances from interferences in the specimen, but also be able to increase their concentrations relative to the co-extracted matrix compounds. The extraction process is often time-consuming and labor-demanding, but practical experience displays that the exertion is indispensable because the subsequent analytical steps are based on and benefit from the sample preparation. To be brief, proper extraction increases the chance of a successful analysis of drugs. Because of the large number of pharmacologically relevant compounds and the different questions that needs to be answered in the various fields of analytical toxicology, a universal standard extraction technique does not exist. Optimized extraction procedures are available for many drugs and can be found through a wide-ranging evaluation of the collected works. However, the toxicologist will over and over again be challenged with compounds for which no suitable extraction procedure has been available, compelling the development of an innovative method based on the available data of the target compound. In a most awful circumstance, neither the physicochemical properties of the target compounds nor the composition of the specimen is recognized. Selecting the best extraction procedure for a particular case and, when required, developing a new extraction method that appropriately addresses specific case challenges are crucial abilities of the analytical toxicologist. The choice or development of a proper extraction procedure and the procedure's adaptation to specific cases based on the analytical data of targeted compounds, existing specimens, advantages and disadvantages of the various extraction techniques, and the time and resources at hand are strategic tasks of the analytical toxicologist. Next, we will study about the fundamentals of drug extraction. The isolation of the compounds of interest from the biological matrix is important for their efficacious detection, identification and quantification. The approach applied and the effort devoted in the development of an extraction procedure depends not only on the physicochemical properties and the expected concentration of the target compounds, but also on the nature of the specimen and the available equipment in the analytical laboratory. In order to develop an effective extraction procedure, some essential principles must be deliberated. with high extraction efficiency for the target compound, it is imaginable to generate a complete analytical procedure with adequate complete recovery. A complete procedure includes the sample pretreatment extraction, fractionation, purification, evaporation, chromatographic separation, detection, identification, and the quantitative determination. The inclusive recovery can be calculated as the percentage of the analyte response after the sample workup compared with that of a solution containing the analyte at a concentration corresponding to the complete recovery. For the quantitative determination of the analyte, and in order to monitor the whole procedure when biological specimens are extracted, an internal standard has to be added at the earliest possible stage. The internal standard must simulate the physicochemical properties of the analyte as thoroughly as possible and must follow the target compound through the entire process of extraction and subsequent analytical steps in order to compensate for any loss of the target compound. Next is the different methods of drug extraction. Here we will start with the conventional methods of extraction. Starting first is microdiffusion. Microdiffusion is a convenient and popular method that facilitates toxicants, that is gaseous and volatiles in blood, urine and gastric aspirates to be isolated, detected and determined. This is done by Conway microdiffusion. Next is UGC method. The solution obtained from the wet digestion process is tested by this method. 1 ml of the solution is taken into a UGC apparatus. 2 pellets of pure zinc metal are put into it. 5 ml of dilute sulfuric acid is poured over the contents. The evolved gas is purified by passing over lead acetate paper so as to absorb the hydrogen sulfide gas and is reacted finally with mercury chloride test paper. Next here is the Marsh-Berzelius method. Electrolytically Marsh-Berzelius test is done over conventional zinc sulfuric acid method for the evolution of nascent hydrogen, as these reagents are often contaminated with arsenic, etc. Scanty materials like burnt bones, hair and nail peelings or scrapings containing the minute traces of arsenic, and for the testing of feeble traces of arsenic present as a natural constituent in tissues, the Marsh test appears to be the only reliable technique available. Next here is distillation. The process involves heating a sample of liquid to convert it into vapors, which is then allowed to flow in another location, where it is cooled, condensing it back into a liquid. Various modifications of the basic distillation process are used for specific purposes such as steam distillation, fractional distillation, distillation under reduced pressure, sweep co-distillation, etc. Solvent extraction. The theory of solid-liquid extraction is that when a solid material comes in contact with a solvent, the soluble components in the solid material move to the solvent. Thus, the solvent extraction of plant material results in the mass transfer of soluble active principles to the solvent, and this takes place in the concentration gradient. The rate of mass transfer decreases as the concentration of the active principle in the solvent increases. until the equilibrium is reached, that is the concentration of active principle in the solid material and the solvent are the same. After that, there will be no longer be a mass transfer of the active principle from the plant material to the solvent. Since mass transfers of the active principle depends on the solubility of the solvent, on heating the sample, it can increase the mass transfer. Besides, if the solvent is in equilibrium with the plant material, then it is replaced with a fresh solvent and the concentration gradient is changed. Next, we will study about the contemporary methods of extraction. Starting with first is the cold aqueous percolation. The powdered material is macerated with water and then poured into a tall column. Cold water is added until the powdered material is completely immersed. It is also allowed to stand for 24 hours so that the water-soluble ingredients achieve equilibrium in water. The enriched aqueous extract is concentrated in multiple effect evaporators to a precise concentration. Some diluents and excipients are added to this concentrated extract, which is then ready for medicinal use. Next is the chromatographic methods. Chromatography involves the separation of substances based on their relative affinity for the two phases, that is the stationary phase and the mobile phase. Substances which have higher affinity for a mobile phase are moved or carried along with it and are thus separated from those with higher affinities for the stationary phase. Thus, the toxicants in molecular mixtures may be separated conveniently under different chromatographic methods and the operating conditions in a particular chromatography. There are different controlling parameters, that is the nature of toxicant, mobile and stationary phase and the temperature. Next is supercritical fluid extraction. The supercritical fluid or the SCFS are gradually substituting the organic solvents, example N-hexane, dichloromethane, chloroform and so on, and that are conventionally used in the industrial extraction operations because of the regulatory and environmental processes or the pressures on the hydrocarbon and the ozone depleting emissions. Most of the presently available solvent-free extraction systems utilize carbon dioxide, which is in general considered as safe for solvent-free extraction process. The fundamental steps that involved in SFE are: first is the liquid carbon dioxide is forced into the supercritical state by regulating its temperature and pressure. The supercritical carbon dioxide has the solvent power and extracts predominantly lipophilic and volatile compounds. The gaseous carbon dioxide returns to the carbon dioxide tank. After a full round, the new extraction starts with flowing carbon dioxide. Next, here comes the solid phase extraction process. The old observation in the extraction of drug by adsorption on solid materials, that is fluorosil, a synthetic magnesium silicate or activated charcoal, and the selective elution of solvents thereafter has given rise to solid phase extraction method. Analyte concentration may often be achieved more easily with SPE than the liquid-liquid extraction, as use of SPE column to concentrate an analyte from the solvent extract may provide a quicker and possibly safer alternative to solvent evaporation. A major advantage of SPE is that the batch processing can be simplified. Further feature when screening for unknown is that A range of analyte can be extracted simultaneously, although this may create problem if analyte of a single component is required. Moreover, the SPE columns are expensive and it may not be possible to retain very water-soluble analyte. Next is the solid phase micro extraction. The solid phase micro extraction, or the SPME, is an extraction technique for organic compounds in aqueous samples in which the analytes are adsorbed directly from the sample onto a fused silica fiber that is coated with an appropriate stationary phase. When the fiber is inserted into the sample, the analyte portion from the sample matrix enters the stationary phase until equilibrium is reached. The fiber is then inserted into the injection port of a gas chromatograph or GC, where it is heated and the analytes are rapidly desorbed thermally into a capillary GC column for the analysis. Next extraction process is the micellar extraction. Micellar extraction is a special type of extraction procedure or technique that appears to be unique in the separation of drugs, plant poisons and pesticides in biological matrices or viscera. In the extraction of active constituents that has been mentioned here, the micellar environment of surfactant of different classes is employed. Surfactant, or the surface active agents at a particular concentration in a solution, are known as the critical micellar concentration, or the CMC form, micelle, or the association colloid. At this concentration or above, marked changes in the properties, that is viscosity, conductance and the electrical conductance, are exhibited. Next is ultrasound assisted solvent extraction. This is an improved maceration technique where the extraction is expedited by the use of ultrasound. The plant powder is placed in a vial and the vial is placed in an ultrasonic bath, and ultrasound is used to induce a mechanical stress on the cells through the production of cavitations in the sample. The cellular breakdown increases the solubilization of metabolites in the solvent and increases the extraction yields. It is mostly used for the initial extraction of a small amount of material. Next is the headspace trapping. The headspace method is especially suitable for the very fast separation of volatile components in complex biological matrices, especially blood in mass liquor and prohibition law related cases. This method has the advantage of avoiding the risk of contamination of non-volatile components, which may be eliminated due to online of the analysis by gas chromatography or GC. The principle underlying the headspace analysis is that in a sealed vial at the constant temperature, equilibrium is established between the volatile components of a liquid sample and the gas phase over it. After allowing the time for equilibrium, a portion of the headspace may be withdrawn one by one from vials using a gas-tight syringe and injected to GC for on-line analysis. Next is Soxhlet extraction. This method is appropriate for both preliminary and the majority extraction. The plant powder is placed in a cellulose thimble in an extraction chamber, which is placed on top of a collecting flask below a reflex condenser. A suitable solvent is then added to the flask and the setup is heated under reflux. When a certain level of condensed solvent has accumulated in the cap, it is siphoned into the flask below. The chief advantage of Soxhlet extraction is that it is a continuous process. Now I will summarize all that we have studied in this module. The process of drug extraction is the principal step in the qualitative and quantitative analysis of drugs from complex biological specimens. The isolation of the compounds of interest from the biological matrix is important for their efficacious detection, identification, and quantification. A complete procedure includes sample pretreatment, extraction, fractionation, purification, evaporation, chromatographic separation, detection, identification, and the quantitative determination. Because of the large number of pharmacologically relevant compounds and the different questions that needs to be answered in the fields of analytical toxicology, a universal standard extraction technique does not exist. The extraction process is often time consuming and labor demanding, but practical experience displays that the exertion is indispensable because the subsequent analytical steps are based on and are benefited from the sample preparation. The choice or development of a proper extraction procedure and the procedure's adaptation to specific cases based on the analytical data of the targeted compounds, existing specimens, advantages and the disadvantages of the various extraction procedures.